Welcome to Barry's In the Interim podcast, where we explore the cutting edge of innovative clinical trial design for the pharmaceutical and medical industries, and so much more. Let's dive in.
Welcome, everybody, to In the Interim. I'm Scott Barry, your host, and I have my usual partner in statistics, my partner in crime, Kert Viele, is here to join me. Welcome back to In the Interim, Kert.
Thanks, Scott.
So we have an interesting topic, and part of it is the failure of AI. That's not the topic. But Kert and I both looked everywhere to see if there's a name for this, to see what AI said of this, and we can't find a name for this. So it's a little bit unnamed. So I am calling it the uncertainty of novelty. The AI threw that out as something, and we'll talk about what this is. And the alternative name of this is something, and I know Kert has a daughter that's in college, and I have a son that's in college, and we learn everything about what's cool in the world from our children, and we learn slang from them, and we've picked up things like dude and bro, and they have specific meanings, and you have to use them in the right particular way. But my son uses the phrase when something just seems awkward, he says, "What are we doing here?" And so it's just that phrase and the way he phrases it. So it's a little bit of mixing uncertainty of novelty and what are we doing here? Okay, so what are we doing here? We are talking about when novelty, and I don't like to use the word innovation because it's almost self-glossing it as positive. But when you're doing new things, you're doing something novel, there are hurdles, and there's people that look at the approach and they point out issues with the novelty. And the interesting thing is those issues exist, and they're maybe worse in the thing you did before that wasn't novel. But yet this is pointed out as these, and it might be barriers that are put on novelty that aren't put on what we do every day. So what are we doing here? I'll jump in the first example, and Kert and I are going to present over the years examples of this. And we want to be really careful here that we're not being critical. Kert and I both have a great deal of respect and appreciation for regulators, for example, journal editors, people who do this. But there are times, these are people who generally provide these hurdles or what about this, what about that, and they show it. So they're going to show up in examples today. But we it's not only regulators, but they'll show up in our examples, and we don't want to come across it at all. So I remember
Scott, we could be upfront that we agree with regulators and journal editors probably more often than our colleague Ed Gilbert.
Yes. A topic for a different day.
Yeah.
So I was working with a sponsor, and we designed. I'll throw out an example. So example number one, we were building a trial, and it was in oncology. And it was a combination therapy compared to standard of care. And the combination was adding experimental agent A to standard of care. Is that better than standard of care? And we were looking at overall response rate as an endpoint. This is looking at the tumor having a 50% reduction, and there are characteristics to this, but that's the endpoint. And the question is, what would happen on standard of care? We actually had data on other trials that had presented ORR rates for standard of care. And we used that in a Bayesian model where we proposed to enroll 3-to-1 to the combination therapy that included the experimental agent and 1 to the standard of care, and use Bayesian borrowing to estimate the standard of care. And the historical data was about a 15% ORR rate. And we proposed this to the agency, so a randomized trial in that. Now, it was back in the old days with some of these FDA meetings, sponsors asked you to physically be located with them, the pharmaceutical company. So I flew to the sponsor, was located there for a telephone call on the design. Now we do this on virtual meetings, and you're not forced to fly to there. Then these were not even flying to DC for the meeting. So I was hammered, as you guys, by the regulators on this design. I was in this. So their complaint was that, okay, you do this borrowing of 15% on historical. Maybe the modern day average of standard of care comparing to your new arm is more like 30%. And by borrowing from the 15% rate, we were doing dynamic borrowing. So yes, type 1 error would be inflated. If the truth is now 30 for the standard of care and we're borrowing from 15, you get inflated type 1 error. Dynamic borrowing is great because it sort of it lowers the rate of type 1 error lowers, and if it gets even higher, it decreases. Beautiful. I thought it was beautiful. And I was crushed by the FDA statistician saying, you're inflating type 1 error. You can't do this. Okay. And I'm there sitting at a table with the sponsor. And so they came back and said, our suggestion to you is that you run a single arm trial of your combination and compare to a historical rate of 15% and show a statistical significant improvement to the 15%, that would be sufficient and a good trial. And so I'm sitting there with the sponsor, and I'm shook a little bit to this. And of course, this is a beautiful thing for the sponsor. They perceive this to be an easier road for them to go. And meanwhile, I want to jump up and down and say, wait a minute, this doesn't make any sense. And we'll come back to this not making sense. But of course, I have to say absolutely nothing. I have to be quiet and say absolutely nothing in all of this. So of course, the interesting thing is, by running a single arm trial to a number, nobody ever asks, what's your type 1 error if that number is wrong? But by borrowing from that same number and enrolling new standard of cares, a novel thing to do, they ask the question, what's the type 1 error if in truth it's 30%? Their solution they came up with, of course, if the current state of nature is that it's 30%, the type 1 error is much, much, much higher in what they asked the sponsor to do. It's much worse than the novel approach. But the novel approach really asks you to investigate that question, and yet it's a better answer for them if that's the concern, but yet, oh, no, we can't do the novel thing. So a little bit of my son's sort of, what are we doing here?
I think that's a little, so I wrote on LinkedIn that an article on can statisticians change reality? And, in effect, that's what's being done here. Type 1 error, if you say, are you making a false conclusion if the null is true? You had protection if the null was 15%. So you had type 1 error control. But the real scientific null you want to test is whether the control equals the treatment, and you're not enrolling control. You don't have protection to that. You have worse protection than what you proposed. We were both in a session at RSIW where Telba was presenting, and she had an example where she proposed it was a non-inferiority trial, 2.5 point non-inferiority margin, and the same issue. You inflate type 1 error if you're borrowing data that's not sufficiently on point, usual problem. But the regulatory solution to this was, okay, we don't want you to borrow, but we will let you use a non-inferiority margin of 3. So in effect, this is changing the goalpost, it's changing reality. You're saying, okay, if you're going to borrow, we think 2.6 down is a bad drug. But if you're not going to borrow, we'll think 2.6 is a good drug in this case. I mean, if your real goal is, I don't want to approve the drug with 2.5, if your NI margin's 3, your type 1 error for that question is inflated. It's inflated everywhere, not just in certain places.
So the 2.5 in the first one with borrowing has a certain type 1 error that can be larger. And by saying 3%, that 2.5 with the 3% has a higher type 1 error than with the borrowing it would have been in that scenario. But somehow there's a comfort in the usual sorts of thing and not the novelty.
Yeah, it's something we've done before, and there's comfort with that. And I have some sympathy for that, but of course it causes us a lot of frustration at times.
Mm-hmm. Okay, so you have another example.
So yeah, so this was several years ago. We're going to switch to enrichment designs, basket trials. So we were presenting it was in psychiatry, and it was a drug that they were interested in testing it in an indication, but that indication could be divided up into 4 groups. And so we proposed an enrichment trial where the 4 groups were in a 2x2 factorial. So we would start looking about, does it work in the top row of that table or the left column or whatever the case may be. We were going to try to personalize where the drug worked and where it didn't. And the problem with this is the standard problem. We were going to use a hierarchical model in order to do this. I should be not upfront. I should be clear. The reason that we can't do separate trials in the 4 groups is it's enormous. We'd have to run at least 4 times the size of the trial. Here it's probably more like 6 because some of the groups are small. So we were going to use a hierarchical model in order to.
But also you have the expectation that the effect might have some similarity in the 4 group. So it's not just cutting the corner. Yep.
Yeah, this isn't random 4 groups. This is the possibility of separate effects, but the expectation they'll be similar. So anyway, the issue when you do this kind of borrowing is there's always a concern. Suppose it works in 3 of the groups and it doesn't work in the 4th. You borrow, you bring that 4th group up, you inflate the type 1 error. So now you've got, instead of having a 2.5% type 1 error, there's this 4% or 8% chance that you're going to incorrectly bring along that last group. So you're going to have this chance of 25% of the population being wrong. So we went back and forth on this, and eventually what happened is all of the enrichment was completely removed, and they ran a pooled analysis. So the pooled analysis, as you were saying in your example, it's worse in terms of this kind of error. So if you run in and the drug actually works in 3 groups and not the 4th, if you pool the type 1 error 70, 80%, you're almost guaranteed to carry it along. And it's one of those situations we've said that now the null makes the implicit assumption about reality that everyone is equal. And we don't think about those type 1 errors. I got jaded after this instance enough to go, it's almost better to do a pooled analysis and do all your subgroups on sensitivities where, everybody's like, well, it's all post hoc and nothing's type 1 error controlled.
Mm. Yeah, so you were proposing a method for that same group that you were criticized for that did better than what the final solution was. So this is a really interesting issue as we get into understanding more about disease, understanding more about populations, potential for heterogeneity of effect. And I remember going to the FDA when we went with the GBM Agile platform trial, and we were looking at potential subgroups of oncology. And we presented this problem where we were borrowing on it. And it's a usual thing. So we don't want to be too critical about people. Initially, this reaction that, by looking at this question, you have to control type 1 error in every possible group. And I thought Lissa Lavange, who was at the FDA at the time, was fantastic in this. She said the FDA approves drugs all the time that there's somebody in that indication that doesn't benefit. And we can't possibly understand that every single person in that population benefits. But yet we do a pooled analysis and a mean effect and we approve it. So the question is, what's a type 1 error if you're going into your example of 4 populations and you end up with the decision that it works in 3, and the truth is that it works in 2, but not the 3rd? Have you committed a type 1 error? And she said, for that particular example, we should count it as a type 1 error if you make an approval and it doesn't benefit any of the groups. But if there's a population in there that it benefits, now this is a mixed result. We want to worry about the likelihood of a mixed result, and we wanted to have that. But to label that as a type 1 error, she didn't think that was a type 1 error, so that was mixed. So we had to do strong control of type 1 as she defined it. And I thought that at the time was an amazing reaction to novel designs that were new, asking new questions and understanding the breadth of things, what the errors were we were always doing, and was able to apply that to a novel scenario.
And I do think this remains an active research area, and I think we don't have a conclusion on this. But the notion of we're used to things like we will tolerate a 2.5% chance of missing the right answer for everybody, approving the drug when it doesn't work for everybody. That's the standard prototypical Stat 200 example. So the question is, what happens when there's a 2.6% chance of only missing it for 20% of the population? Is that a worse error? I mean, I'd argue it's less of an error, but we need to actually, this needs to be worked out. And then we need a metric that works in this case.
Yeah. Okay, we may come back to enrichment trials. We think that the draft ICHE 20 is misleading and imposing a restriction on them. Maybe I shouldn't do that. So do you want to describe what potentially the ICHE 20 says, since we're on enrichment trials?
All right. Well, since you went there, so the other interesting aspect when you do enrichment is there's a statement in the guidance, and it's in a number of guidances. It isn't unique to ICHE 20, but there's a question if you investigate multiple groups and you show an effect in some of the groups, and let's assume it's a strong effect and so on. The question is, what do you have to do with the groups that you're leaving behind? So do you need to show that it doesn't work in those groups? And if you do have to show that, the question is then what is the burden of proof? Do you have to demonstrate the null is true, which is obviously hard? And Scott, I'm going to let you, I know this is near and dear to your current heart.
Yeah. And we got a response from regulators quoting the draft ICHE 20 that it says for enrichment trials that you have to show that the effect is significantly larger in the groups you're taking forward than the ones you're not. And that burden has never been placed on a sponsor before. If you run a phase 2 trial and you're looking at and you decide these are my inclusion-exclusion criteria for phase 3, you are never asked to demonstrate that people you're not enrolling have different benefit to the drug. It would be an enormous burden. It would mean you'd never run an enrichment trial. But the standard development pathway, you don't ever have to do that. But now, because you're addressing the potential differential, which, by the way, is better for patients, this isn't a bad thing to be doing. You now have a higher burden by doing that than if you don't do it. And of course, what it does is it pushes you to do the same thing, which is worse.
And essentially, to come back to what you're saying, this is, if you have an inclusion right here, I'm going to enroll people over 50. They never ask you to show that it doesn't work for a 45-year-old. It's just not part of the. I do think there's a road here that the question, it is nice to show there's a differential effect. And all of these designs, they're aimed at showing a differential effect. The question is, it's vague on saying what is that standard of evidence. There's certainly a level of evidence that the design provides. We just don't know what the bar is. And significance is way too high.
Yep. Or having to prove that the drug doesn't work. Because many times in an enrichment trial, you're enrolling a trial and you don't enroll groups where the effect is, it's probably positive, but likely small. Very severe patients, you might have a small benefit, but they're hard to get to, and largely there's little difference, or very healthy, then everybody does well. So it's just a statistical thing not to enroll that, where you likely have a benefit, but to prove you don't, we would all grind to a halt. A similar example is shifting gears to another one that came up when a new trial came to the forefront of platform trials. So a platform trial is a trial where in a single trial you enroll multiple experimental arms. And so a simple platform trial is we enroll experimental arms A, B, and C, and they're all compared to a common control. Equal randomization, simple trial, but in the same trial, there's A, B, and C comparing to a control. Initially, when we were doing designs like this, and others were doing designs like this, the initial reaction was you have to control the type 1 error for A, adjusting for the fact that you're testing B and C. And so instead of 2.5% type 1 error for A, which is the standard, you have to split that because you're investigating B and C. And we kind of understand how we get here. We use terms like experiment-wide type 1 error when we're looking at old trials that only addressed one arm or addressed versions of one treatment, like doses. And so the parallel to a platform trial that's looking at A, B, and C is we run 3 separate trials, one of A against control, one of B against control, and one of C against control. The standard is they all get 0.025 error. But by putting them in the same trial, you have to adjust for the other ones. And by the way, if you add a fourth, you might now have to adjust it even further in this scenario. Is a burden put on that's never put on it, the trial in the other circumstance, and by the way, would crush platform trials never to be run. The nice thing about this example is there was that initial reaction to it, regulators, journals, review people, things that I think we've largely come to the recognition that, oh, this is something we've always done. This is a burden, that this is not something we should place upon an additional burden to this novelty that we don't place in other scenarios. And guidance documents at the FDA are reflective of this. Now, there are interesting cases where these things are all the same mechanism of action, or if they happen to be doses, we all recognize that that's different science. But in the case where there are 3 different experimental arms, this was new, and that was what was thought, but it completely differed from what we normally do.
And I can actually see putting a multiplicity penalty on separate trials. I mean, we talk about all the time, a certain the amyloid hypothesis in Alzheimer's, which is somewhat evolved recently. So this isn't as good an example as it was 5 years ago. But the notion of, does one trial out of 30 investigating the same mechanism, what does that mean? The key issue here is if you're going to do one thing for separate trials, you should be doing the same thing in the platform, whatever it is.
And we all think that actually we do a poor job of analyzing across trials. And part of it is that all the characteristics we look at are within trial type 1 error, within trial power and all that. But, and a call for a different day is whether we'd be better off with one large trial or 2 small, adequate and well-controlled trials. But other things come up in that circumstance.
I've seen bizarre ones where they've said a company should only have a certain amount of error, and it's sort of, well, the company needs to spin off a unit in order to actually test another drug and things like that. It gets a little strange.
Yeah, it gets strange because it probably means I have no type 1 error left, and I need to retire. Yep.
It's the only reason we hire.
Yep. Okay. You have a response adaptive randomization scenario.
Oh, yes, I do. So this is somewhat related to the platform trials example. It involves multiple arms. And so that's the specifics. Kind of the general topic here is a lot of the ethical discussions that come up in adaptive trials, where we're looking at the data at an interim. So someone knows what the data look like. And you often get into ethical discussions of, have you broken equipoise? So an RAR example, you look at the data, one arm's doing great, the other two arms are doing poorly. You increase allocation to the one, you decrease allocation to the two that are doing worse. And they're not doing bad enough to fail, they're just doing worse. And then the question is, well, that's unethical because you have evidence that the good drug is better. So have you broken equipoise? And this is always, first off, certainly have some sympathy for the argument. Should you treat the next patient in as best you can according to what you know? The counter argument to that, of course, is we can't just always use the best arm. We know that we don't learn efficiently when that happens. If I just said, after the first patient, I'll pick the best arm and always give that, I won't get the right answer at the end. So I have to balance this learning and randomization somehow. So, but the thing that this always seems odd about to me is if I were to not look at the data at all. So I haven't looked at the data. I'm not running an interim. I could be sitting in a situation where I have the same data set where X is doing better than Y and Z, and I might continue to allocate 1 to 1 to 1. I continue equal allocation. So in that example, I give more of Y and Z to patients in the trial than I would doing RAR. So the patients in the trial suffer. They're going to have a worse result. I also know that RAR is more likely to get me the right answer, has more power, has better chance of picking the right dose if it's done correctly. And so I end up, by looking at the data, I treat the patients outside the trial better. So in effect, the knowledge that I have, I'm being told that if I use it, by using it, I treat the patients in the trial better, I treat the patients outside the trial better. Everybody wins, but if I do it, but somehow that's unethical, whereas if I blindly hurt people, that's okay. And I just, so to be provocative here, this is the example I use when I want to get a, kind of a rise out of people, I'll go, in most cases, if you do something if you refuse to look at information and cause harm, that's often considered negligence. It couldn't be a virtue here.
Yeah. And I've heard the same thing, that if at an interim there's an 80% chance dose 2 is better than dose 1, you shouldn't give dose 1 anymore. And the act of doing the interim and learning that answer means you can't give that anymore. But if you don't do the interim, it's okay to do fixed randomization. The knowledge is there, the data is the same, but somehow ignorance is bliss, and it's okay in that circumstance. Response adaptive randomization, even doing interims, brings out a lot of these scenarios where the perception that what you now need to do because there was a look that's made imposes things that it's okay to just ignore it in other circumstances. Okay, I'm going to jump on an analysis method one. This is near and dear to my heart. If you were at RISW a couple weeks ago, I probably shouldn't timestamp this, but I talked about this a little bit. So in analyzing an ordinal outcome, we have developed a utility weight to an ordinal outcome called the modified Rankin score. There are 7 outcomes of the modified Rankin score, from 0, which is perfect neurological status, integer values up to 6, which is dead, neurologically dead. And there's differential level of disability from 1, 2, 3, 4, 5, up to 6. And we have done multiple studies that look at the impact of the different states, and we weight them relative to economic burden and patient preference. They're almost identical. We've created a weight to that. And we've labeled it the utility weighted MRS. And we ran this in a trial design actually was tied to an enrichment trial design. And we ran the trial design, the Dawn trial. It was the primary endpoint in the Dawn trial. And the trial was incredibly successful. Endovascular therapy is an amazing therapy. It's awesome. We proposed this, and we got within the journal Stroke, the investigators from Mr. Clean wrote in about the utility weighted MRS. And there were a number of complaints, but the biggest complaint they had was, you can't do that because you're using a relative weight of the outcomes when not everybody in the population agrees to those weights. There were other things largely that we wrote back to them, and they came back. And the single point, and lots of people have this criticism, that you're doing weights of those outcomes and not everybody in the population is the same. They even looked at EQ5D for their trial, Mr. Clean trial, which also showed benefit of endovascular therapy, and showed that the utility that people gave the state 1, 2, 3, there was variance. Not everybody said the same thing. So you can't do this weighting system for it. And their solution was, you should do a proportional odds model. Many others say you need to do a dichotomization of the ordinal outcome. If you do a dichotomization of the ordinal outcome, you are imposing utilities on those states that are 1, 1, 1, 0, 0, 0, 0. And you are imposing that everybody in that population has those weights. If you do a proportional odds model, you are imposing weights on the outcomes. The weights actually depend on prevalence, but you're imposing a single weight to each of those outcomes for everybody in the population. It's a little implicit in that scenario. And this was a group, very smart group, do wonderful trials in this, but it was this reaction that you can't do this because you're imposing everybody in the population agrees to that, so do this, which does exactly that same thing. It imposes a weight on those values for everybody in the population. It violates the single thing that they thought was the worst reason exactly, and somehow that's okay, where what we did was bad. And largely it was because we wrote down what the weights were, where you can implicitly hide it behind a statistical assumption if you don't write it down.
And I think this comes up, we've talked about this offline a lot, where there's a lot of situations where statisticians, we're implicitly making a lot of clinical judgments in trials that I don't want to do. I mean, if I form a win ratio statistic, there are implied utilities in those levels. So if death is the first level and time on ventilation is the second level, you're making an implicit decision of how much extra death will compensate for how much extra mechanical ventilation. It's in there, but it's not written down explicitly, and that's in the statistics, and it's not thought about. Whereas if I were to write this out as an explicit, here is the utility for different patient paths, then it becomes a major argument. And I would much rather have clinicians and patients making those kind of judgments explicitly, even if imperfect, than me doing it implicitly and no one sees it.
Yeah. A great example of that is the CAFS analysis of ALS trials, where you do function, ALS, FRS, and death. And you do a non-parametric test of those because they don't want to weight death and function. It's hard. So they do this non-parametric analysis, which weights death and function implicitly through a statistical assumption. It's just not written down, and somehow that's okay. Where I, as you say, I'd much rather have this done, even if not perfectly, done and explicit by people who should be working on that, not a statistical assumption to those. Yeah. All right. I think we have managed not to insult anybody by saying,
I hope not. We certainly didn't intend to.
What are we doing here? I think it's the natural thing of when you're doing something different. I'll call it novel. When you're doing novel things, you're doing new things, you start to address questions that you never addressed with the old comfortable one. And all of a sudden you worry about that issue and realize, the thing I was doing before actually had exactly this same issue. We just didn't really worry about it, the comfort of that.
Yeah, I think we definitely, whenever you do something new, we should always be thinking in depth, tear it apart. What are the risks? All of that makes sense. But at the end of the day, what we really end up doing is defining a new metric. What are we considering to be the problem that we're trying to solve? And the new methods don't have to be perfect about that. They need to be better than what existed before. And in order to assess that, you need a common framework. And so you don't want to switch it where the novel method is addressed on this metric and the old method is addressed on something different. It's not a good comparison.
Mm-hmm. Mm-hmm. I actually think that's one of the values of clinical trial simulation. I think we can in depth look at a deep set of operating characteristics that are very hard to look at in other scenarios. And so apples to apples comparison of approaches and picking better ones with explicit performance and goals written down and shown, I think is very helpful. All right. Our time in the interim is done for today, Kert. A really cool topic. And we see examples of this and appreciate you coming in the interim.
Thank you.
All right, everybody. Thank you for joining us in the interim. Until the next one.